So as an example for replication, we could have this double strand of DNA where we have this sequence of bases, A, C, G, A, G, G on the first strand, and its complementary base pairs, T, 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 G, C, T, C, C on the complementary pairs. And if it was going to replicate, the next thing that would happen is that strand, which is actually a twisted ladder, would untangle, unzip, and you would have this right here. And so now we just have this original strand, AAA, CG, AGG over here, and its complementary strand, TTT, GC, TCC on the other side. It's just split open, same original old strands. And we have eaten, and so we're going to have a bunch of bases floating around in the nucleus as well where this is happening. And so now it's just a question of what might float in. There's a lot of things that could float in next to this A, but which one would match? Well, the only thing that would match an A is a T. And so we might have that float in and be next to the A. And what would be next to the next A? Well, again, the only thing that will actually stick there is a T. And so the T will float in and we'll get another T. What about that third A? Well, again, we're going to get a T. That's going to match that third A. And we can continue this where the next thing that matches C, if a C floats in there, will it stick? No, C doesn't match with C. We would have to get a G. A G would have to float in and match with that C. You can see I'm not getting a very straight DNA here. I'm trying. And the next thing that would match a G, well, what matches a G? If a T floats in there, will it stick? No, only a C floating in is going to actually make it in there. And so what might match with an A? How about this T? This T will stick there, so let's put that in there, right? And what matches with G? Well, if a G floats in there, will it stick? No, only a C is going to fit, right? And what about to this G? Well, again, G is only going to match with C, so a C could float in there and match, and we're going to have that first strand finished. How does that strand, I know it's a little sloppy, but how does that strand compare to our original? It's identical. And what might match in here with this T? Only an A, right? So let's bring that A in there, and that's going to match. How about to the next T? What's going to fit? Will a C fit in there? Heck no. We got to get another A, and an A is going to fit in there and match up with that T. And what's going to match with that third T? Can we put a G in there? No. See? No. We're going to have to pick up an A and plop that in there. How about with the G? That's right. We're going to put our C in there. And if we continue matching C's to G's and T's to A's, we are going, will that fit? No, I picked up the wrong one. What's going to match with that C? We need a G. It doesn't want to go. Let's give that another shot. Let's see. There's our G. What's going to match with that T? Now we can get that A in there. And a G will match with that C. And finally, the last C, we're going to get a G. Now, I know I was a sloppy DNA maker, but how do these two new strands compare to the old strand? They're identical. The only thing that's different is that the original outer strand, the part I have in black, is the old nucleotides. The new ones that match are the blue ones on the inside, and we now have two identical strands simply because the DNA has loose hydrogen, weak hydrogen bonds and can unzip and zip back up. And because we have base pairing where only A's go T's and C's go G's. And so you can have DNA in the original cell, and when it copies, make two identical copies that are exactly the same as the original. And that's how a cell manages to keep the DNA the same so that every single cell in your body gets identical DNA.